Hi, all. I'm Josh Lovell, one of the application specialists at Teledyne ISCO, and thank you for joining us today. Today's webinar is a unique experience as we have the opportunity of having Dr. Chris Coxon present about his research into the discovery of new anti-migraine peptides and how the use of Teledyne ISCO's ComboFlash NextGen series and ReadyStep Gold material have helped advance his research. So I'll be moderating today with questions and also assisting if you guys have any questions regarding the ComboFlash systems at the end or anything like that. But we're really lucky to have Dr. Coxon today. About him, Dr. Chris Coxon is an associate professor in synthetic chemistry and a bicentennial research leader in the School of Engineering and Physical Sciences at Heriot-Watt University in the UK. His research program focuses on peptide drug discovery and using peptides as tools for biology. And specifically, his team is interested in tackling neurological and neurodegenerative diseases. He completed his PhD in 2010 in the development of novel inhibitors of protein kinases at the Northern Institute for Cancer Research at Newcastle University, before having postdoctoral appointments in chemical biology and peptide chemistry at Durham University in 2010 to 2013. He previously held academic positions at Durham University and Liverpool John Moores University between 2013 and 19. And additionally, in addition to his academic role, he's now a director and co-founder of Pepmotech Limited. Thank you. Professor Coxon, would you like to introduce Professor Coxon?
Thanks very much, Josh, and thanks to Teledyne ISCO for arranging this webinar. I'm delighted to be here and delighted to share with everyone just some brief parts of our research. So thanks everyone for tuning in wherever you're listening from. As Josh said, I'm Chris Coxon. I'm a medicinal chemist, and I just want to tell you really a little bit about our anti-migraine drug discovery program. I'll also tell you a little bit about how we design our peptides, how we make the peptides using solid phase peptide synthesis, and then how we've used flash chromatography methods to purify peptides when ordinarily we might have used preparative HPLC. And we find that this has really allowed us to purify larger amounts of peptides in a shorter space of time. But before we get on to the science, I'd just like to show you where our lab is. And we're based at Heriot-Watt University in the School of Engineering and Physical Sciences. And this is in Edinburgh, which is the capital city of Scotland. A really beautiful city if you ever get the chance to visit. And my lab's based in the William Perkin building, named after the first professor of chemistry at Heriot-Watt University. So over recent years, my lab have gained an interest in diseases of the central nervous system. And really this is motivated by the fact that in the UK, there are around about 15 million or about 1 in 6 people who suffer from at least one neurological condition. Due to the aging population, this is likely to become a larger problem in future years. And so we're interested in finding ways to understand and tackle these problems. In particular, we're interested in Parkinson's disease, which has around about 120,000 sufferers in England alone, and in migraine. And migraine, I think there's an astonishing number of sufferers with around about 8 million sufferers just in England. And for context, the population of the UK is about 67 million. So this is a large proportion. Now, our goal as a research group is really to try to improve the provision of new knowledge and new treatments for tackling diseases of the central nervous system, including some of those I've just mentioned. So specifically, our group are interested in migraine, which is what we'll be talking about today, but also in chronic pain, and there are some overlaps there. We have an interest in targeting and treating brain cancers, And unconventionally, we're actually trying to use peptides to do this, which I'm sure most people will know presents a few problems. One of those problems is trying to get drugs of any kind into the brain and bypassing the blood-brain barrier. And it certainly is a challenge. We're working with some collaborators to try to develop some novel nanotechnology-based approaches that can transport drugs across the blood-brain barrier to treat a variety of neurological and neurodegenerative diseases. And we've published a couple of reviews on this subject recently. We're also interested in diseases that are typically characterized by protein misfolding. So these are diseases, generally speaking, dementia-related illnesses like Alzheimer's and Parkinson's. And again, we're interested in using fluorinated tools to understand protein folding, and there's a review on this topic just here. And you'll see some elements of this today. So the key themes in our research group are peptide chemistry, organofluorine chemistry, using thiols as reactive handles to modify peptides and proteins, and also improving the sustainability of, in particular, peptide chemistry. Okay, so the main focus of today's talk is on migraine. And for anyone who doesn't suffer from migraine, those lucky people, I think this picture quite nicely sums up how a migraine might feel. It certainly looks quite painful. However, those who do suffer from migraine will be able to tell you that migraine is not simply a bad headache, as it's often misunderstood to be. In fact, migraine is classified as a neurovascular disorder. It usually presents as a really intense headache just on one side of the head, and it's often associated with this premonition phase, or this phase before the headache arrives, in which the sufferer suffers from altered sensory perception, maybe altered vision, and this is sometimes called an aura, and it might present as flashing lights or things appearing the wrong color, or zigzag lines is quite a common aura. And the headache phase of migraine is usually accompanied by nausea, vomiting, real sensitivity to light and to sound. And if this doesn't sound unpleasant enough, it can actually last up to 3 days in certain sufferers. And people with chronic migraine can actually suffer very regular bouts of migraine attacks. And it can in fact be quite disabling. Unfortunately for some, the prevalence of migraine increases typically with age up to a certain point, and women tend to be disproportionately affected. To come back to the point I made before, I was very surprised to hear that the World Health Organization considers migraine to be the third most disabling disease in the world, and that might surprise a few people. It's estimated that around about 14% of the world's population are actually affected. And just in the UK, that causes around about 25 million work days to be lost due to people taking absence through illness. And ultimately, that costs the UK economy around about £3.5 billion per year in lost productivity. So I think you'll agree that finding good treatments for migraine is actually a bit of a priority. Now, one symptom that's often associated with migraine is this feeling of a buildup of pressure inside the head. And unsurprisingly, one of the earliest approaches to attempt to treat migraine was actually to drill a hole in the sufferer's head to release the pressure. Now, I guess you'll agree that that probably wasn't very successful, and I'm sure you'll agree that the migraine was probably preferable to a hole in the head. Now, more recently, we've noticed a link between vasodilation, so that's the dilation of blood vessels in the brain, and this being linked to pain and also to migraine. And this has led to the discovery that, for example, this natural product called ergotamine, which is a product of this fungus, ergot, that grows on rye crops. Ergotamine is able to actually relieve some of the symptoms of migraine. And the only problem with ergotamine is the eagle-eyed amongst you should notice that this structure bears a striking resemblance to the drug of abuse, LSD. So ergotamine actually has some unwanted psychoactive effects as well, which makes this potentially a bit of a toxic drug, although it is actually still used for migraine treatment as a nasal spray. Now, the way this probably works is, I've highlighted a part of this molecule in red, but I'll just emphasize it here. There's a tryptamine motif in this molecule, which closely resembles serotonin, or 5-hydroxytryptamine. And this molecule probably works by mimicking serotonin and binding to and activating the serotonin receptors. And this is a family of receptors, the 5-HT family of receptors, that control a huge number of functions, one of which is vasoconstriction. And so by activating that receptor or those receptors, that triggers vasoconstriction and results in the relief of some of the symptoms of migraine. And then that led to the rational development of drugs of the triptan class, typified by sumatriptan, that's shown on the right here, during the 1990s. And these are reasonably useful drugs, but unfortunately, they're typically only around about 60% effective, which means that a large proportion of people taking the triptans won't see the benefit that they would ideally like. One of the main drawbacks of the triptans is that they're actually contraindicated in people with cardiovascular disease. So that again reduces the number of the population who can see a benefit from this drug. Also, a consideration is that some of these agents can actually induce different headaches. They can cause nausea, dizziness, hallucinations, and actually with prolonged use can in some cases exacerbate the symptoms of migraine. And if that doesn't convince you, it's recommended if you're taking sumatriptan that you shouldn't drink red wine, coffee, or eat chocolate or cheese, and that's presumably due to tryptamine content, I guess. So I think you'll hopefully agree that we would like to develop new methods to treat migraine, and ideally methods that directly deal with the cause of migraine. So more recently, researchers have discovered that a neuropeptide, the calcitonin gene-related peptide, or CGRP for short, is causally related to migraine. So CGRP is a 37 amino acid neuropeptide, and it comes in two forms in humans. These are the primary sequences for the alpha form, which is the one that we are interested in, and then the beta form. The beta form has a couple of minor amino acid substitutions, but the functional significance of those is not really well known. So CGRP is a potent vasodilator, and it's also linked to the transmission of pain. So we might see how this could be involved in migraine. However, there are two very important studies that clearly implicated CGRP in migraine pathophysiology. The first of which was that during a migraine attack, it was possible to detect high levels of CGRP or metabolites of CGRP in the blood or urine of the migraine sufferer. And in fact, interestingly, if that person was then treated with sumatriptan, that could actually reduce the levels of CGRP and also relieve the symptoms of migraine. A second study actually found that if you take a migraine sufferer and inject them with CGRP, which sounds quite cruel to me, you can induce migraine-like headaches. So really what we have here is a cause and effect. So let's have a think about how CGRP works from a chemistry point of view. Well, for simplicity, I've shown the CGRP receptor as this blue surface here, and actually it's more than one part, but for the sake of today's talk, we'll call this the calcitonin gene-related peptide receptor, or CGRP receptor. Now, in this red or orange color, we can see the CGRP peptide actually bound to the receptor. And this has been a really useful study that was published about 3 years ago from some other researchers. And this has led to us better understanding how CGRP binds to its receptor, because having that knowledge allows us to, in theory, design molecules that should mimic CGRP and bind in preference to the receptor, and therefore prevent the binding of CGRP and prevent the activation of the receptor and prevent the switching on of various pathways that lead to the pain transmission that causes migraine. So to some extent, this has already been attempted, and there's been some success in this area using small molecules. There have been a number of small molecule CGRP antagonists that have made it to clinical trials, some of which are ongoing. However, the first small molecule CGRP antagonist, ubrogepant, shown in green here, was actually approved by the US FDA in 2019 for acute treatment of migraine with or without aura. Now, a second class of medicines to try to target CGRP in migraine are the monoclonal antibody class. And one example of this is Heriot-Watt. And Heriot-Watt was approved in 2018 as a once-monthly subcutaneous injection to prevent migraine. And the interesting thing here is that the monoclonal antibodies have a long half-life in the blood, so they can be taken as preventatives. So whilst Heriot-Watt targets the CGRP receptor and blocks the binding of the peptide, much similar to the way that the small molecules work, there are monoclonal antibodies undergoing clinical trials now that actually target the peptide itself. So we might have a really good range of new treatments to treat migraine. However, we feel that peptides offer another solution and maybe a new way to treat migraine. So peptides typically combine the best features of some of the other classes of the small molecules and the monoclonal antibodies. They can often have very high target selectivity and target affinity. But on the other hand, they can have low immunogenicity and low toxicity. And from a manufacturing point of view, some peptides are relatively straightforward to manufacture and relatively low cost compared with some of the biologic alternatives. So I suppose at this point I need to tell you our vision of peptide medicines. Well, in November 2020, we conducted a study that showed there were now over 100 approved peptide pharmaceutical agents, and these are used for either therapeutic or diagnostic purposes. And you can see, if you look at this chart, that since 1948, when insulin was first approved to treat diabetes, there's been a steady increase in the cumulative number of peptide approvals. Now, the peptide drugs market is expected to increase to a value around $50 billion US by 2027. And that's actually double where we were in 2018. So this is quite a sharp increase, and it's largely down to new technologies and new ways of discovering peptide drugs. Now, of course, peptides have advantages and disadvantages. We've mentioned some advantages. Typically, the main disadvantage is the metabolic liabilities and the challenges of improving the pharmacokinetic properties of peptides. So we found that of the approved peptides, only about 1 in 8 are actually what we call heterologous or designed peptides, whilst around about 7 out of 8 are either natural products or analogues of natural products, so closely related versions of the natural product that have been augmented in some way to improve their properties. And I think that probably the reason that there are so few heterologous peptides is probably because there are no clear rules for the successful design of a peptide drug. And we're still very much learning about what we need to do here. So as a way to start to address this problem, Vera and Paolo from my lab recently developed a freely available database of regulatory approved peptide therapeutics and diagnostics. And we call this Pep30. And this is accessible on the web, and the web address is actually shown here. And I'd encourage anyone who's interested in developing peptide drugs just to have a look here. We've been able to catalog all of the approvals and group them by certain properties. Now, using this database and some statistical analysis, we've been able to try to start to unpick some of the structural trends in approved peptides to help us design peptides that are more likely to be approved, albeit a long way into the future. So for example, we've discovered that most of the approved peptides are fairly small in that they contain fewer than 10 amino acids. They typically have a molecular weight below 2,000. There are a number of ways that the C-terminus or N-terminus can be modified, some more successful than others, or at least more widely represented in the approved list of drugs. One important observation was that around about 50% of peptides contain at least one macrocycle. And really interestingly, we thought, is that generally speaking, these macrocycles contain somewhere between 5 and 8 amino acids. And there are a variety of ways, again, some more widely represented than others, for making the macrocycle. And whilst I can't give too many details here today, we've tried to use some of these details to inform the design of our new compounds that we're designing to target, to mimic CGRP and antagonize that interaction with this CGRP receptor. Okay, so now let's look at the key features of the CGRP peptide as a starting point for the design of our antagonists. So CGRP contains 3 main parts. We've got this N-terminal hairpin-like motif, which contains a disulfide bridge. We then have a central alpha helical domain and then a C-terminal hairpin motif as well. And the C-terminal hairpin contains an amide at the C-terminus. And that's really important, as you'll see quite soon. One interesting modification is that if we remove the N-terminal hairpin motif, we obtain the compound that is the amino acids 8 to 37 of CGRP. And actually, this can bind to the receptor with very high affinity, but it can't activate the receptor. And this is a really important discovery because this compound actually therefore can work as an antagonist. It can bind to the receptor, it can't switch it on, but it occupies the receptor and prevents the binding of the full CGRP peptide. So this is a very decent antagonist. However, unfortunately, it's not very drug-like. So this molecule can be broken down further into helix mimetics or some C-terminal hairpin mimetics. And actually, we're working on both parts of this as we speak. But for today's talk, I just want to tell you some brief details about work we've done on this C-terminal section. Okay, so let's have a look at the structure of the C-terminal domain. And it's important to note that we're not the first people to consider making modifications on this part of the peptide to design new antagonists. So let's have a look at some of the modifications that are known in the literature. So this is amino acid 23 all the way through to 37. And it is known that this can be truncated somewhat. Some of these amino acids can be replaced. Some of them are not tolerant to replacement, and this is probably because the peptide has to adopt a particular conformation. And it's known that removal or replacement of valine 32 and glycine 33 actually causes a significant loss in binding affinity for the receptor. So this seems to be affecting the conformation. I mentioned earlier that we need a C-terminal amide, and this is found in the peptide. If we remove that, we see a significant loss in activity. However, the parts of this molecule that we're particularly interested in, certainly for today's talk, are the ones I've shown here. So first of all, this proline 29 position, if that's replaced with an alanine, this results in about a 400-fold reduction in binding affinity. So this is really crucial for the biological activity. Similarly, if this serine here is replaced with a proline, we actually see a beneficial 10-fold increase in affinity. So it's quite clear that having prolines in these two positions highlighted in blue is very useful. The lysine here can be replaced often with aromatic type amino acids to improve affinity as well. Whilst if we remove this C-terminal phenylalanine, we completely lose affinity. And you'll see some of these emerge later on. So let's have a quick look at our approach to developing new anti-migraine peptides. Well, first of all, we typically start by, we gain our inspiration by looking at the parent structure of CGRP. And then we try to design, sometimes using computational approaches, and sometimes just old-fashioned medicinal chemistry know-how, to design molecules that can mimic this and might therefore act as an antagonist. So this is a ligand-based design approach. We're then able to go ahead and synthesize peptides using solid phase peptide synthesis, which you'll see in more detail in a moment. We then perform a series of studies such as circular dichroism, for example, to ensure that the peptide adopts the right conformation, or even studying stability in blood serum, which is an important consideration for a peptide drug. We try to evaluate the biological activity of these compounds using a commercial functional assay, which measures the production of cyclic AMP upon binding of CGRP to neuronal cells, and how that can be reduced by competition from an antagonist. Compounds that have shown some promise at this stage have then been evaluated in some fairly preliminary in vivo experiments. And we've actually, as I'll show you in a few slides' time, we've actually obtained some pretty promising in vivo biological activity. The next stage is really to explore the efficacy of these compounds in sheep. And that seems a bit of a strange choice, but sheep have a very similar nasal vasculature to humans. And our plan is to deliver these peptides as a nasal, as a nasal spray, because we think we can access the parts of the body we need quite quickly in that approach. So we're also working with Professor Gillian Hutchin at LJMU in Liverpool to develop some novel formulation approaches for these peptides so that we can safely deliver them via a nasal spray. And we're also working with some companies to develop nasal spray devices. So we typically make our peptides using microwave-assisted solid phase peptide synthesis, and this usually involves using the CEM Liberty Blue peptide synthesizer. So we've got room for the 20 natural amino acids, plus extra room for some other reagents or unnatural amino acids, and we use this for making peptidomimetics quite often. The reason for making peptidomimetics is this typically helps us find new ways to either find new biological activities or improve biological activity, or even to increase the stability of the drug. So finally, the key feature here really is the microwave reactor, and this allows us to rapidly synthesize peptides with about a 4-minute cycle time. So we're typically looking at somewhere around 2 hours, for example, to synthesize the peptides we're interested in. So we build our peptides using a rink amide linker. And this is important because the amine here affords a C-terminal amide. And we've seen already that this is essential for the biological activity. We use a protide resin. Again, we buy these from CEM. The protide resin is a combination of polyethylene glycol and polystyrene. And generally, for the longer peptides that we synthesize, we use a lower loading capacity on the resin. So solid phase peptide synthesis generally consists of this iterative cycle of removal of FMOC protecting groups on the terminus of the peptide, or even in the first coupling step, removal of the FMOC from the resin linker. And we generally do this using piperidine, a base. Then after copious washing to remove any excess piperidine, we then couple our next amino acid, which is usually protected on the N terminus with an FMOC protecting group. Again, base sensitive. So once we've coupled this, we can go around again and remove the FMOC group and attach the next amino acid. Now, the couplings are typically performed using diisopropyl carbodiimide and oximer, and the coupling times are around about 2 minutes per coupling at 90 degrees, although we sometimes have to adjust that depending on which amino acids we're using and whether they're prone to racemization, for example. So once we've prepared the peptide, what we should have is a resin. This sort of circle represents the resin. Onto that, via a nitrogen, we have the peptide attached. And this peptide has usually, certain amino acids have side chains that need to be protected to avoid any unwanted reactions. And that's what we've represented by these gray balls here. Now, when we treat with trifluoroacetic acid, we cleave the peptide from the resin, and we also cleave these side chain protecting groups simultaneously. We also need to include something, a scavenger like triisopropyl silane, as a way to mop up any reactive species that are generated during that process. And then if things have gone well, we should have a nice clean peptide with a C terminus that is the amide. And then I can show you an example here of a peptide that contains 17 amino acids. 4 of these amino acids needed to be double coupled to ensure they were successfully attached with no gaps in the peptide. And we achieved the synthesis of this peptide in about 1 hour 45 minutes on the standard loading resin on 0.1 millimole scale. And you can see from the LCMS analysis here that actually this is a very clean peptide product. So when things work well, this is the kind of result we can expect. In some cases, that doesn't always work. And this is the subject of the application note that's in the bottom right of the screen here. So in one project, we've been trying to make some peptoids. And peptoids are peptide mimetics in which the alpha carbon substituent is relocated onto an adjacent nitrogen from an amide bond. So we have synthesized these compounds and found, after cleavage and analysis, we found that we've obtained the compound we want, but we have almost a one-to-one mixture of the desired compound and an unwanted side product. And the unwanted side product we were able to determine came from an incomplete coupling reaction. So this is where that cycle of solid phase peptide synthesis broke down briefly, and we weren't able to get full coupling. And we call this a deletion product. So ordinarily, if we wanted to try to purify these peptides to separate the desired peptide from the undesired, we would use preparative HPLC. And this is the standard trick to use in peptide synthesis. However, on this occasion, we thought we would have a go of doing some C18 reversed phase purifications using our next-gen flash system. And what we found, as you can see on the screen here, we were able to quite cleanly separate the undesired compound, the deletion, from the pure desired compound. And I think you'll agree that these look like pretty successful separations. Now, in general, we've been able to use this quite a lot, and we found that the flash system can offer fairly quick purifications of generally larger quantities of peptide. And of course, it's dependent on the resolution between the two signals, but in general, we found this to be a pretty successful way to purify our peptides and peptidomimetics. This method obviously needed some optimization. And you can see here that when we use a standard ReadySep C18 reversed phase column and subject this to a water methanol gradient going from 5 to 95%, in fact, it's 5 to 100%, in fact, of methanol. With a low sample loading, we were able to achieve some reasonable resolution of the two compounds. Unfortunately, when we scaled that up to use a larger sample loading, you can see here that the resolution was somewhat lost. And that was a slight limitation. However, we then decided to compare this with the ReadySep gold C18 columns. So these have a smaller particle size and spherical particles. And what you can see, if you compare the left-hand spectra chromatograms rather with the right-hand ones, you can see that with low sample loading, we achieve pretty good signal resolution. And actually, it's just as good with the higher sample loading. So this has allowed us to purify quite large quantities of our peptides, certainly more than enough for our biological assays, in just a single run by flash chromatography. And actually, the advantage of this is that the single run is a lot quicker than using the prep HPLC, which in our experience typically takes several runs to obtain enough material. But I would also just like to show you the comparison prep HPLC chromatograms. And you can see that it is entirely possible to separate these by prep HPLC. They're over slightly longer times, and actually we were loading slightly less peptide material as well. So I feel that in general, the flash approach using the gold C18 columns was just as good in our experience as using prep HPLC. So in fact, it's been simpler because we've had this in our own lab rather than having to share a system, and it's been faster really. So actually, we typically use this now to purify our peptides. And so I've got another couple of examples really just to highlight how successful this has been. Here's a peptide that is by the UV chromatogram reasonably clean, but we do have some impurities. But when we look at the mass spec, we can see that it's, to be honest, full of junk that we need to try to remove. So when we perform a flash separation using the gold columns, the gold C18 columns, you can see that we can spot two distinct compounds, but we get some pretty reasonable resolution. And then after flash chromatography, the HPLC analysis shows that we've got a pretty clean peptide by UV, and then mass spec analysis also shows that we've got essentially just the one component under that peak. And again, these are separated in fairly short times with reasonable sample loading. A slightly more complicated or slightly messier example is a peptide here. Again, this has a number of close running impurities, and ordinarily we would probably think we would need PrEP HPLC to successfully purify this compound. But as it turns out, again, it's messy by mass spec. As it turns out, we were able to obtain a nice clean product by flash chromatography using gold C18 columns, and by UV, this is a perfectly clean peptide, and by mass spec, also acceptably clean. Okay, so this is Vera, who's done a lot of the work I've just shown you, and here she is looking delighted next to her combi flash system, and she is smiling under the mask, I can promise that. Okay, so what I'd like to do now is come back to the design of some of these antagonists. So as a reminder, this represents the C-terminal peptide sequence of CGRP. And this itself has a reasonable antagonist potency. But if you remember, I mentioned the importance of these residues in blue and how when they're both proline, this affords a benefit. We have pretty potent antagonists. And we're interested in the role of the prolines here. Now, proline is interesting as an amino acid because it's able to adopt both cis and trans conformations relatively easily compared with normal amino acids. And by adopting these different conformations, this can be used to stabilize certain conformations of the peptide, or even secondary structures of a peptide. And so our group also have an interest in fluorine chemistry, which I mentioned at the start. And what we found is, or what we wanted to do, was to replace proline, which has a relatively low preference for the cis conformer, as you can see in this chart, with different fluorinated prolines, which are known to have differing preferences for cis and trans conformers. And we wanted to do that really to probe the importance of the conformation of these prolines on biological activity. So to do this, we took one of our lead compounds, and I can't give you too much more detail on this, I'm afraid. We're waiting on a patent to be published before we can talk in any real detail about this. However, I can tell you that it contains two prolines, and we decided to replace each of these prolines one at a time, and then in combination with the different fluorinated prolines. And I've just color-coded the biological activities here just for ease, really. So the green represents a very low nanomolar potency, antagonist potency, whilst a red, unfortunately, represents something that's largely inactive. So if we replace with the 4R fluoroproline, the 4S fluoroproline, and the 4,4-difluoroproline, we can see that these have different effects on the biological activity. We can see that the 4S fluoroproline is really the only one that's tolerated here, and this is the one that affords a slightly higher preference for the cis conformer. If we look at the proline 34 position and replace with 4R, 4S, or 4,4-difluoroproline again, what we see is that in general, any modification here is not well tolerated. In fact, the only one that's moderately tolerated is the 4,4-difluoroproline, which actually has a similar preference for cis and trans proportions to proline itself. Unsurprisingly, when we start to combine these fluoroprolines, we see that we generally just lose activity. So we think this is interesting because, okay, we haven't enhanced the potency of these compounds, but what we have done is learn about the sensitivity of these positions to modification. And in particular, we think that the proline 29 position favors a cis conformation, and we're looking at ways to stabilize that now. Whilst we think that anything you do to the 34 position proline is generally detrimental unless it's proline. Okay, so let's go back to the full sequence of CGRP, and that's shown here. This is the human alpha form of CGRP. Remember, we've got the N-terminal disulfide bridged hairpin. We've got this area in the middle that is alpha helical, and then we've got this C-terminal motif that adopts a hairpin-like structure that we've just seen, and also we have a C-terminal amide. Now, I mentioned earlier that we can remove this N-terminal part, and we don't really lose any affinity, but we lose the ability to activate the receptor, so that's good news. We can further truncate this peptide to obtain this peptide, the amino acids 27 to 37. And this affords a modest antagonist of CGRP. However, as you've just seen, if we replace the serine at position 34 with proline, and also I mentioned earlier, we can replace the lysine with phenylalanine, we can obtain now a really potent antagonist. And actually a further modification is removal of this C-terminal phenylalanine in this compound here results in a complete loss of binding affinity and a loss of potency. So then we've gone on to test these compounds, and what you can see from this series of lines here in the different colors is a dose response curve, and we see that increasing dose causes increased inhibition. So we have a dose-related increase in biological activity. And this demonstrates that we're out-competing CGRP. So it's functioning as a competitive antagonist. Now, I mentioned earlier that one of the things that we want to do is to develop these for nasal delivery. And one way we're hoping to do that is by formulating these compounds as a dry powder in low molecular weight chitosan. And we're trying to develop microparticles for nasal delivery. We've been able to do that quite successfully. And what I'd like to show you is the same dose response curve that compares the unformulated peptide in the red line, or the square points, with the formulated, which is in green, or the triangles. And the key message to take from that is that the formulated peptide actually has identical biological activity. So we're happy that our formulation, first of all, doesn't preclude the biological activity, but also it doesn't do anything unexpected. So we're happy with that. The next job was to show that these compounds actually have in vivo activity in a rodent model. So the rodent model we've used is one that measures plasma protein extravasation as a model for vasodilation. So how this works is the rodent model is injected with a dye, Evans blue, and we measure the amount of leakage of that dye from the blood vessels into the surrounding tissue. And this is a model for vasodilation. It's expected to be increased with increased vasodilation. So what we can see here is that in the first cohort, animals treated with CGRP have a higher level of plasma protein extravasation, or a higher amount of leakage of the Evans blue dye compared with those treated with just a control, or PBS control. Now in cohort 2, we see again, two animals treated, one with just CGRP and one with the lead antagonist, or this compound shown at the top. And what you should be able to see is that there is a marked reduction in vasodilation. So that indicates that we are having some biological activity. And what's interesting as well is that when we, instead of using the potent antagonist, if we use the negative control compound that lacks this C-terminal phenylalanine, we see that there is now no reduction in vasodilation. So that confirms that what we see in the cell-based assay translates to the in vivo model, and that these compounds are indeed inducing vasodilation, vasoconstriction. Okay, so we have since gone on and developed some new compounds, and I'm afraid I can't show any sequences or structures here while we wait for our patent to be published. What I can show you is that these are a subset of the compounds we've developed, and what you can see is there are a range of different biological activities and different antagonist potencies. And the reason that that's of value is that we've been able to now elucidate some of the key structure-activity relationships. But also we've discovered compounds such as the one shown at the bottom here, 024, which has comparable antagonist potency to the compound I've just shown you, but in fact has far superior pharmacokinetic properties. And actually, we measure the pharmacokinetic properties, in particular by looking at blood serum stability. And of course, this is one of the real drawbacks of peptide-based medicines, that they generally suffer from a very short half-life once they reach the blood. And so we have to make special efforts to try to enhance that stability. So typically what we do is we take a solution of our peptide in 25% human serum, blood serum, from a commercial source, and we incubate that for a defined set of time. We then stop the reaction by adding trichloroacetic acid, and this causes the precipitation of the proteins and the proteases that would normally chop up the peptide. We centrifuge this to remove any of the precipitated proteins, and then analyze the supernatant solution using analytical HPLC. And that's what we see in this middle figure here. So this is the HPLC chromatogram for one of these particular peptides over a period of, 1 hour. And what you can see is that we start off with one single peptide, and over time, we see this is quite rapidly degraded until after 60 minutes, there's essentially none of this left. But it's converted quite cleanly into one new metabolite. And this new product, well, it's the result of a single cleavage point by a single protease, we think. And actually, by looking at the mass spectrometry data, we're able to identify exactly where this cleavage site is, and identify the nature of these compounds. And by doing so, we're able to then rationally design where we need to make specific modifications to enhance the stability. And I can show you here, if we plot this is one series of compounds, if we plot the remaining intact peptide on the y-axis versus time, and really we're just integrating these signals in the HPLC, we can see that one particular modification has made quite a significant improvement and significantly extended the protease resistance. Another compound, again, that I can't show you a structure of, but this is one of our lead compounds, we've also shown has biological activity in a similar in vivo model to the one you saw earlier. So this work was conducted in collaboration with Professor Helias in Hungary. And what we do here is we take a rodent and inject into one paw and the other one either a control, which is just the vehicle, or the antagonist. And so we've got antagonist on the right, we've got vehicle on the left, and then 30 minutes later, we inject with CGRP peptide. This is the full-length CGRP, and we know from the earlier work that that triggers migraine in migraine sufferers. We also know it's a potent vasodilator. So what we're looking for here is a reduction in vasodilation, which is we're measuring using laser speckle contrast imaging. And you can actually see there's good evidence here that the treated pore has significantly less vasodilation than the untreated pore. So we've got further evidence that we have in vivo biological activity and that this is consistent with the biological assays and the structure-activity relationships. We have started to do some work in sheep, and we found that there is no toxicity, but this is via intravenous and intranasal administration. But this is very early work, so I wouldn't like to say too much more about that. So what I've really done over the course of this talk is give you a very brief flavor of some of the work ongoing in our lab, some of the things we're interested in particular drug design using peptides, a little bit of fluorine chemistry, and some new methods for purifying peptides, which is becoming an increasingly important part of our work. So to summarize what we've seen, we've used a ligand-based approach to design some new potent antagonists of CGRP. So these are based on the CGRP parent peptide to a large degree. We've shown how we've used microwave-assisted solid-phase peptide synthesis to prepare the peptides, and they've then been successfully purified using flash chromatography in reversed phase using our next-gen system. And actually, this, as far as we can see, appears to be comparable with PrEP HPLC. We've developed some serum-stable analogs, and actually we're really pleased to see that we have in vivo biological activity. And so we're currently working on the formulation parts of this project in order to make these peptides able to be administered nasally. And we're also working on some new peptidomimetic modalities. So we have up our sleeves a range of very potent compounds. I would love to share them all with you today, but unfortunately we don't have time for that, and I'm also not allowed to tell you everything. But hopefully that's been of interest. If you are still interested and would like to find out more, then I can recommend reading one of our papers here that was the first we've published on the migraine project. We're waiting for others that are due to come out very soon. If you're interested in the discovery of peptide drugs in general, then please read our Drug Discovery Today article that summarizes some of the trends that we've discovered and also links through to this database I mentioned, Pep30. If you're interested in the flash chromatography side, then I really encourage you to read our application note in collaboration with Teledyne ISCO, and also look out for upcoming publications. And if you want to know the latest goings on in the lab, please check out our Twitter page. So this is our group as things look in 2021. Unfortunately, our group photograph had to be via Twitter and via Zoom rather. And in particular, I'd like to thank Vera, who's done the bulk of the work that you've seen today. In addition to Vera, though, this is the rest of the migraine team. We've got Professor Gillian Hutchin, who's in charge of our formulation and pharmaceutics arm of the project. Professor Dave Kendall, who's in charge of the pharmacology, and in fact runs Innovipharm, the company that we're working very closely with. And then we have Vicky and Paddy, who are the postdocs who have done quite a lot of work on this, on formulation and peptide synthesis, respectively. And with that, I'll just thank again the organizers, thank Teledyne ISCO for the opportunity to give this talk today. I've really enjoyed the opportunity to talk about some of our research. I hope you've enjoyed it too, and I'm happy to take any questions. Thank you.
Thank you, Chris. That was an excellent talk. Got a lot of great questions here to ask you from the audience. So if you guys have any other questions, feel free to submit them via the QA function on Zoom there, and we'll try and get them answered. If we don't get to them, we will get them answered via email. First question here. Really enjoying your talk here. Question was, why don't you consider acetonitrile instead of methanol in the mobile phase? They suggest maybe it is superior to methanol, but you also have several hydrophobic residues there. So,
yes, I guess, yeah, thanks for the question. It's a really good point. And in fact, in some cases, we do actually use acetonitrile in place of methanol. I guess really our first point of call is to use methanol just because it's cheaper, to be honest. But in many cases, we sometimes have to screen both to get some to look for the sort of optimal separation. Thanks.
Yeah, that makes sense. Always want to go with the cheaper solvent when possible. So if you get sufficient, separation, that's the way to go. So, let's see, another question here. They understand that the data is still unpublished, but the modification to enhance the peptide stability, was that done by using a non-proteinogenic amino acid like AIB or maybe a D-amino acid?
Right. Yeah, another good question. So we've tried a number of things really, and I suppose I can say that in some cases we have used non-proteinogenic amino acids. In some cases we have looked at switching the stereochemistry. In other ways we've looked at just modifying the sequence using other natural amino acids in order to improve the stability. We've actually worked out where the main cleavage site is, and so we can just pretty much chop and change around there to improve the stability.
Okay, fantastic. Another question here, and this is in regards to the purification. So have you been able to use your reverse phase flash chromatography to purify larger peptides such as CGRP, which is about 37 amino acid residues?
Yeah, thanks for the question. So we have attempted this. Unfortunately, we haven't really been able to purify this as we would like. And I think the main problem is actually the synthesis. We generate too many byproducts in this synthesis. I think if we could improve the synthesis and reduce the number of deletions, then the flash chromatography approach would probably work quite well. In fact, at the moment, I have to admit, we buy the CGRP just because it's just relatively cheap, and we have a good supplier.
That's fair. Let's see here. Next question there. Have you been able to get any information about how the peptide specific interactions with the receptor at all yet?
Right, that's good. At the moment, we don't have any structural biology work, and that is something we're desperately trying to get access to. That would be really valuable. At the moment, we've done a little bit of computational work, mainly docking the peptide or looking at modified and then optimized structures that we've played with in software packages, but nothing that we would consider to be really informative, I guess. No, that would be certainly something we aim to do.
For sure. I got 3 more questions here. Let's see. What kind of pressures do you like to use for the difficult purifications?
So I have to be honest, I don't really know the answer. I guess, I don't know, Josh, maybe you have a better idea than me. I'm not entirely sure what pressures we reach.
Yeah.
We don't do anything to particularly control that, is all I would say.
Yeah, I guess probably just the jump from flash to prep there. So flash normally, probably under the conditions you were running, you were at lower flow rates, but in the reverse phase systems, you'd probably be under 100 psi pressure. But when you jump to that prep HPLC, now you're in the 2000 to 3000 or more psi, depending on what kind of stationary phase you're using, and particle size there. So I assume, the more difficult purifications, like you were talking about with the larger peptides, with the synthesis, you have more impurities. So you probably need that added resolution by moving up to the prep HPLC to get those impurities out. I would think.
Yeah, yeah.
And
That
so
makes sense.
just naturally that kind of generates a little bit more back pressure from the smaller particle size.
Yeah, yeah. Okay.
Two more here. So do you synthesize your fluoroprolines in-house?
We have done, but in actual fact, it seems to take too long, really. So we just buy them in. I have to say they're relatively expensive, but we just have to weigh up what costs more, the time of the student or the or the reagents.
Right.
And they're quite expensive, unfortunately.
Need to get some undergrad interns.
Yeah.
Last question here. For making the chimeras, did you try to use chloroacetic acid and potassium iodide?
No, a good question. We actually just used bromoacetic acid. I guess reasonably similar. We didn't use the potassium iodide, and however, the couplings worked pretty well, to be honest. There is an example in there, of course, where we have that fairly significant deletion, although that was not standard. We don't often see a deletion like that. It just made for a good example for us to try to play around with some of the flash purification methods, really.
Sure. Okay, it looks like all the questions I have. Chris, I want to thank you again. This is an excellent talk and really good engagement from the audience. So obviously a lot of people were interested there, so that's great. But appreciate your time and sharing how the next gen and the ReadyStep Gold material helped help you reach your goals and your lab's goals and make that easier for you guys.
Thank you. Really appreciate your time, and thanks everyone for listening.